What is the scientific core of RESIST?
We are testing an overarching theory on the role of tolerances to environmental stressors, of dispersal and of biotic interactions for degradation and recovery trajectories. And we are testing this for components of the entire food web.
Near natural water bodies are vital to biodiversity and also to humans. But many are now degraded by human activities. These two states, near natural or degraded, illustrate our research focus.
We selected two catchments, the Emscher-Boyer catchment in an urban area that is recovering from severe sewage pollution since 20 years. And the Kinzig in Hessen, a countryside area representing a typical Central European landscape.
We explore the mechanisms and processes of transitions between these two states. This allows for insights into stream ecosystem functions and their resistance and resilience to multiple stressors.
During degradation and recovery, many stressors such as hydromorphological alteration, temperature and salinity act simultaneously.
What can be observed, for example, after restoration measures, is an asymmetric response of communities, leading to different functionality and community composition compared to the pre-degradation status. A main target of RESIST is to understand the effects of stressor interactions on all organisms groups, from viruses and bacteria to fish and parasites, and also on selected ecosystem functions. A major strength of RESIST is the link of 5 central approaches that integrate the different individual research projects.
We used the aquaflow mesocosm systems to study the effect of multiple stressors under laboratory conditions.
The extreme experiment is one of the central experiments within our collaborative research center.
With these methods, we can combine the standardization and high replications with the field natural conditions. So with these advantages, we can use it for the in-depth statistical analysis.
Well, extreme is really important because it's actually best of both worlds. It has the whole replication and control that any lab experiment would do, but it's done in the field, so it's also a field experiment.
The concepts from lab and semi-field studies were validated in our model catchments.
We analyzed more than 34 terabytes of sequencing data, not only for single organisms, but for the whole community, and this we did by sequencing metatranscriptomes and metabarcoding data.
Because of the sheer amount of data that we generated within RESIST, we needed an overarching project that organized all that respective data under one roof. A tremendous challenge for our data management project and our infrastructure was a cyber attack in November 2022. Although it took us months to restore the original data, we now can build our future projects on a stable and secure system.
RESIST has a systematic approach to modeling, aiming to understand the responses of species and communities to environmental variables, to stressors and recovery. And there are some models such as hydrological models, species distribution models, or food web models that were used in the CRC.
We had a very strong synthesis component where we tested the project's main hypothesis with data from all projects, but also with external data.
And we generate some generic insight how river ecosystems react to stressors and also to recovery.
Our goal is to understand and predict the effects of stressors during degradation and recovery on freshwater biodiversity and function. Our doctoral researchers are integrated in the graduate school of RESIST that includes career development plans and non-academic mentors.
The main advantage that I got from the RTG, it's the fact that it's a structured program that equipped me with all the skills that I needed to complete my thesis successfully.
The IRTG had many different workshops and summer schools in which all doctoral students joined, so we could work together on different projects and learn from each other.
As we started in the middle of Corona, it was really important to have my PhD sisters and brothers from the IRTG really from day one to have support and somebody to talk to about science and also the whole personal struggles in the middle of epidemic.
RESIST also contributes to the further scientific or professional development of all members.
During the first phase of RESIST, I got a professorship at the Westphalian University of Applied Sciences, and the CRC contributed a lot to the success of my application.
We're looking back to a successful first phase of RESIST due to the exceptional work of our RESIST team.